as far as Connect goes, if you signed up for the courtesy access and it expired, you should have got an email. And that wasn't from me. That was like automatically generated from Connect, letting you know that it expired and you needed to pay your money now. So for us, let's see, where is our class? Right here. I got an email too telling me that some people's was getting ready to expire. So you'll notice, right, some of you guys need to pay your monies because it says expired. You won't lose any of your work that you did, but you definitely need to pay, and you definitely want to have access to those assignments. So when I go to my assignments page, one of the questions that one of my students had asked me in my previous class, you'll notice that when you click on the assignment, I'm not in student view. Let me go to student view. So the good news is that, 26 of you submitted, right? 5 of you guys are still working on it, and 5 of you guys haven't even started yet. That's kind of scary because that was chapter 1. That was a little while ago, right? You need to get on the ball. So it says assignment attempts remaining unlimited. So I give you guys unlimited number of attempts, and I just randomly set the due date in the afternoon on the day of our test. But obviously you want to do these assignments in preparation for the test. And remember, how did I say to do them? As if they're what? A test or a quiz. No notes, no book. Because how many times can you do this assignment? Unlimited. So don't worry about your points. Worry about what you know and don't know, right? See how much you know the first time without looking at anything. Right? So I think we went through this one together because I had some students that were having a problem with it. I don't remember if I recorded. But you'll notice, even though they don't write a category over here, they're kind of based on categories. So this first one is cell organization, right? So archaea are usually single-celled, where eukarya can be what? Single-cell or multi-celled. So where is that one? Over here. So, and then bacteria are usually single-celled. Right, and then you drag it in. If you're having trouble with all of that, remember this runs on like Flash, I think. So one of the first 0-point assignments I have in Connect is, has animations, has drag and drops like this, so you can make sure that it's not a software issue before you go to do an assignment. But any assignment you do, you can do again. So everyone understand that one? So there are 21 different questions in this one. The point values are there. So you could save and exit it, or you can submit it. It isn't until you submit it that it will let you do it again. Does that make sense? And then, of course, you have access to the ebook. You can even print them if you wanted to. You can have it check your work. Oh, look, report an issue is back. Aha. Remember I complained about that? It had gone away for a while. So like if you have a problem with a question, like something's not doing like it's supposed to, you can click on report an issue. You guys want to finish this one? Why not, right? Good review. Okay, so nucleus is the next, right, category. So no nucleus for bacteria. What about archaea? No nucleus. Where is it? Right here. And what about eukarya? Has, right? That's the defining characteristic of that category, is it not? Yes. Okay, so the next one is dealing with peptidoglycan. What is the peptidoglycan? It's a cell wall. It's an actual chemical composition of a cell wall that's unique to who? No. Just bacteria. Just bacteria. So let's find the peptidoglycan. Here we go. And give it to our bacteria. And then by default, the no goes for our archaea. Okay? And so now we're talking about environments, right? So what are our environmental choices? Found in non-extreme environments. Who's that? That's eukaryotes, right? Remember, we don't like hot and cold and salty. Where archaea, you find them everywhere, but they really like the extremes. So by default, found in all environments. Yep. Okay. So the next one is cell type, right? Prokaryotic structure or not. So who's prokaryotic? Archaea, right? And then by default, not prokaryotic in structure, but what do we call these guys? Eukaryote are all eukaryotes. So this particular question didn't go into any more detail than I did in my lecture, correct? Right? And this falls into a couple of different objectives, right? Being able to distinguish between prokaryotic and eukaryotic cells. And definitely that objective that deals with the 3 domains and knowing their characteristics, right? And then we had a table very similar to this one that was in my lecture. So let's go to the next one. And so this is a multiple choice. I'm going to skip over that. All right. So this one. Yeah, this is the one I want to talk about. Okay. So this one's fun. Complete the sentences, right? So in ancient times, people believed in the theory of what? Spontaneous generation. So we just plug it in there. Which stated that no non-living organisms, no, you're right, living organisms from non-living matter, right? They just poof, spontaneously generated, but we know that not to be true, right? Testing this theory, a series of experiments carried out on rotten meat by, who did the rotten meat with the maggots? Yeah, Francisco Redi. I heard someone say that one. Now, obviously, in Canvas, we don't have the ability to do these drag and drop fancy things, but how else could I ask this question? Could be a matching, right? Could be multiple choice. I won't make you guys type anything in. It's going to be matching, multiple choice, and true and false. Okay? However, more evidence supporting the theory was then generated by who showed that slightly... Yeah, Needham. A few years after Needham, is a clue right there. Blank improved on his experiment by sealing the broths. Lazaro. That's Lazaro. Support of the theory then hypothesized that sealing the flask would not allow the vital force to enter, right? And so blank allowed superheated air to enter boiled flasks of broth, and still no spoilage occurred. Isn't that Pasteur, though? Yeah, but Pasteur, remember the swan neck flask? I think this is what they're referring to, maybe.
Pasteur is the one under it.
Oh, Lazo's the other way?
No, Pasteur.
He's the final one?
Yeah.
Oh, because they don't talk about John and Cohen, which they don't even have on this list. So, I have a feeling I am going to check my work, right? And we're good. But guess what? I'm pretty sure this answer is one of the guys over here we didn't talk about. Is it Theodore? There we go. Schwann. Let's see if we got it right. So you looked it up in the book, didn't you? All right. Did we cover him in lecture? Is he listed on any of my objectives?
No.
Do you need to know him?
Yes.
No. Right? Because he's not on the objectives and he's not covered in lecture. But he's in this Connect assignment because I can't edit him out. Do you see this? But we covered just about all the other guys that were important, right? So it was a good review for you. So this is, I am sorry, I cannot edit out, and I'm not going to add him in either. He's not, well, you know, I'm pretty sure you guys don't want to study every single one that's in the chapter, right?
No.
Okay. So you're okay with this, right? Do you understand from now on, if there is some detail, like an extra guy in this case, that I don't listed on my objectives or in my lecture, do you need to know him? No. But you could quickly look it up in your book, right, so you can get it right on your assignment. Now, if you skipped him the first time, right, you'll get a score, it'll tell you the right answer, and you can go back and do it again and fill it back in. Right? Make sense? I knew I'd find one that was a good example of what I wanted to explain. Okay. Examples are always good. All right, so I'm not going to continue on this assignment because we've got some people who haven't done it yet either. So, but I'm going to go ahead and submit. It's telling me I haven't completed it. It's going to give me a really crappy score, 0.5 out of 10 points. But guess what? I can do it again. And guess what score will count for your final grade in the course? The highest score you get. Okay? The highest score you get is what will count. All right? Does that make sense to everybody? I have the deadline sent for the test because this is going to, and you should have been working on, you should have already completed the first 2 assignments, right? Let me go back to our home. You should have already done chapter 1. You should have already done the gram stain animation, right? We kind of finished up mostly prokaryotes, so you could have done that, but it's okay if you haven't yet. But you definitely want to finish these 2 very soon, right? Because test is Tuesday, okay? So speaking of the test, I just added for you guys, let me go to modules. We're going to use a special program. It's already on the computer at the computer lab that we're going to. And I'll post an announcement and a map, and I'll even have a note here in case you forget and you come here on Tuesday. But we're going to be meeting in Building 1, which is the Hibernia Center, room 108. It's a computer lab on the first floor at the back of the building next to Einstein Bagels, if you've been over that way. You do absolutely need to know your Canvas username, which everybody knows their name, so we're good on that. But your password, right? Some of us, like myself, my computer remembers it for me. You may want to write it down, put it in your cell phone or something like that if it's a complicated one and you think you're going to screw it up under test anxiety conditions. Because the first thing you're going to need to do is log into Canvas. But you're not going to log in using any of the normal web browsers. You're going to use this program called Lockdown Browser. So this is the program right here. You see the icon on my desktop? Right here. Lockdown Browser. So when you go to the computer lab, you're going to find that one on the desktop. You're going to click on it. Right now I can't go to it because I have other programs opened. And that's why it's called Lockdown Browser. And actually right now it's updating, which is probably good. It'll go right to Canvas, and the first thing you'll need to do is log in. I'll update this later. Oh, it's letting me go. Yeah, and see, it's going to tell you you got to close your other programs before you can do anything. And so this is so that you guys can't cheat during the test. I don't want you to close any of my stuff. So you'll be locked into that browser. Right? You won't be able to leave the test. You won't be able to surf the internet. Right? All that good stuff. So that's for testing security in the computer lab. The nice thing, too, about taking your test on the computer is that once everyone's taken the test, I'll make your results available to you. But you have to use the Lockdown Browser to view them. So what I have done is given you guys our Delgado link to the Lockdown Browser. So if you used it for other classes, you might already have the link. So here's the link right here. Right? You can download it to your computer. Even if you have a Mac, you can download it. And this is the old times, but they're probably pretty much the same. So this program is in a few locations on campus. The second floor right up here on the east side. You have to have your student ID to use that lab. And again, they have the icon right on the desktop there. These are their hours. They're probably pretty much the same this semester too. They're closed for a few days during midterms and finals because we proctor the tests for students to take online classes. They have to come in and take their midterm and final. The Hibernia Center, which is where we'll go. Sometimes this lab is not available because instructors reserve it, like I've done for our test on Tuesday, right? So no one else will be able to go in there, just us, because it's been reserved for us. So when you go to download the program, right, there is a little button, like if you have a Mac, right, so you can download the version for Macs. So it doesn't matter which one. So you can download this at home, right, and once I make the test results available, you again close everything on your computer, you click that lockdown browser icon, you'll log on to Canvas, you'll go to your test, and you'll be able to see what ones you got right and what ones you got wrong, what the correct answers are, and what objective that question linked to, right? Because remember, your objectives are your study guide. I take that objective and I pick or write test questions based on that objective. So the thing with that too is, so something like this is an example of one of our objectives, right? So distinguish prokaryotic cells from eukaryotic cells. So here's an example of a multiple choice question that could be on your test. So prokaryotes, and then the answer is C, right? Lack membrane-bound organelles. Well, it really just, for that question, just really focused on prokaryotes, right? But you should also know the flip side is that eukaryotes have a membrane-bound nucleus. So if after the test, if you got it wrong, you see 0 points, right? You're like, oh crap. Notice that the question focused on prokaryotes, right? But the objective that was meant to assess also deals with eukaryotes. So on the final exam, I could ask a different type question. I could ask, all prokaryotic cells are smaller than eukaryotic cells. It's false, right? Okay, just checking. Or I could ask something related to eukaryotes, right? In this case, A and C are correct. And I can do that, by the way. Some of the questions can be multiple correct answers. You'll notice that instead of little circles, there are boxes. And above the question it will say, or within the question it will say, check all that apply. That means you're going to probably check more than one box. Does that make sense? And again, they're boxes, not circles. Circles, one click. Boxes, more than one click possible. All right, so that's what the icon looks like. There's the free thing. So after, you can either check at home if you download this program at home, which is free, or you can go upstairs to the Open Lab or the Hibernia Center when it's open to review your test results. You cannot print your results, but you have access to them whenever you want, pretty much, right? Especially since you can download the program at home. I don't let you print because then you're going to pass out my test and sell my test, and then I have to write really hard tests. Besides, you don't need to print it. You need to write yourself out your own little study guide based on how you did on your test, right? So if you miss that objective, you're going to probably either take out your objective list and highlight that one, make some notes as to, where you went wrong and where you need to improve for the final exam. Any objective that's on a test is a 50-50 chance of being on the final. I don't give you a final exam study guide. I gave you a study guide at the very beginning. They're called your objectives for every single topic. That's where my test questions are going to be based from, is your objectives. I cover them in every single lecture. Right? For the final, I can't give you a study guide. I don't know what you learned or didn't learn from the previous test. You have to do that yourself, but I give you the tools to do that. Does that make sense to you guys? So after test 1, you should already be studying for the final, right? Because you will know what you knew or didn't know. And hopefully, you know, that list is very short, right, as to what you need to review. Because we're all going to get A's on Tuesday, right? I'm totally fine with that. That would make my day. Sure, it would make yours. Okay. Make sense? Any questions at this point?
Is that a practice test that you can access?
No, this is just instructions.
Oh, okay.
This is just instructions.
Also, did you say
As soon as everyone's taken the test. So if everybody shows up Tuesday and takes the test, then Tuesday night, I'll turn it on. That's rarely the case, though. I would probably expect the day or the day after.
Are we meeting,
It happens, though, occasionally.
Are we meeting at the Hibernian?
Mm-hmm. So you're meeting at the Hibernia Center. You don't need a student ID for the Hibernia Center. They'll have an access code that you'll plug in when you sign in. But you do, as I said, need to know your username and password for Canvas because you will be logging into that to take the test. You will know your results the minute you hit the button. You'll know your score once you get done taking the test. You won't know which ones you got right or wrong until I release that, right? But you'll know your score.
And the other question I have is it on Eukaryotes and Prokaryotes? Like, we haven't finished Prokaryotes.
Right. It's on these topics, yes. So this example is very much related to the test, this test 1. So our topics for test 1 are microscopy, microbiology, which was the first one, microbiology, microscopy, prokaryotes, and eukaryotes. Right? Oh, wrong class. That's you guys. So all of Module 1, which we're just about ready to finish up. All right, so microbiology, microscopy, prokaryotes, and eukaryotes. So I have one more lecture to post here. In fact, I'll do that right now. You see the inner workings of Canvas. So 4 topics. There you go. So let's go to today's lecture. And I believe did we finish up about endospores? Yeah, I think we stopped right here. We weren't finished. Okay. So with endospores, remember this, all this terminology sounds like reproduction, but it's not, right? It's a dormant cell type. 2 genuses that you should know are Bacillus and Clostridium. And then one of the rarities is Sporosarcinia, which happens to be a cocci. Unlike Bacillus, which the name implies, what shape is it? Bacillus rod-shaped, right? And Clostridium are also rod-shaped bacteria. So very much reminiscent to the diagram that we see here. So because of that, because typically a lot of your spore formers are rod-shaped bacteria, their location within the mother cell, before the mother cell deteriorates, the endospore, can help in identifying which species of bacteria you're looking at. So if it's right in the middle, it's said to be centrally located. If it's at the end, of course, it's terminal. And if it's not quite smack in the middle, but not all the way at the end, right, it's in between, it's subterminal. So it's not at the end, it's not in the middle. And then sometimes when the spore forms, it's actually quite larger than the mother cell itself. So you actually get this swollen appearance. And again, this one is showing which location for the spore. This is terminal, but they can be swollen central or even subterminal. But a particular species will always form their endospore in the same location inside that cell. So this is very useful in identification. So there are 4 forms of locomotion utilized by bacteria. So notice again, we're focusing on bacteria. So a lot of them have what that allow them to move? Flagella, right? And the flagella on a bacteria spins like a propeller on a boat, right? It has a little hook to it, so it's angled and it rotates. How else can they move? There's one actual shape that allows them to move. It's helical. What's the name we give to those guys that are spirochetes, right? The spirochetes. They corkscrew through their environment because of their axial filaments, right? They can bore through. Some of them have another appendage outside that can help them kind of twitch along. Do you guys remember what that? The fimbriae, yep. And then some of them kind of glide like a snail would because they produce what? Slime. That gliding motility, right? So we have flagella, moves like a propeller, right? Rotates. The spirochetes, they corkscrew through their environment due to their axial filaments. Some of them can glide due to slime production, and some of them can kind of twitch along because of their fimbriae. So if they have the ability to do locomotion, which not all do, right, but a lot of them can move, they'll swim, right, especially through a liquid media. Or if they're on a solid surface, they'll glide or twitch. Or they'll corkscrew. And they're going to head towards chemical attractants and away from chemical repellents. So I'm actually going to go to this animation. That I posted for you guys. Right, so here's this one that shows the chemotaxis. And I'll make it big. But I noticed somebody muted. I'm hoping. That in. Hold on a second. Okay, I wasn't doing anything. Okay, so for this animation, you see this guy swimming around. So he'll orient all his flagella, because he's covered in flagella, to be able to run in one direction. And he's moving those flagella in a particular direction to be able to run or tumble, as you saw. Well, how is it that he knows that good stuff is in that direction or bad stuff that he may want to be heading away from it? Well, what is this embedded in his membrane but a protein? And this is where, I tell my students, pretty much stop listening to this animation because it's going to put you to sleep. You see all these acronyms, right, and all these molecules, right? The take-home message here, let me make sure I pause the correct recording. There we go. Is that this is it says a methyl accepting protein. So this is a protein embedded in their membrane. Now, of course, this is enlarged like gazillion times for be able to explain it. But remember, a membrane is phospholipids and a whole bunch of proteins. A lot of these proteins are receptors. They're sensors for this bacteria to be able to sense its environment. So this one is for a particular type of proteins. And so when they interact with this receptor, it sends a signal inside the cell, right, as to what it is and how to respond. So they're going down to the molecular level, right, saying which molecules are involved in this process. But eventually, what's going to happen? It's going to determine how that propeller, how that flagella moves. Are we going to keep running? Are we going to start tumbling away from this? Is this good or is this bad? Right? And this is how they sense. It's proteins embedded in their membrane. Protein receptors in their membrane are going to sense the environment and determine, is this good, is this bad, am I going to run towards it, or am I going to tumble away from it? You know, so for us, you're sitting in the bedroom and you wake up and then all of a sudden, ooh, I smell bacon. I don't know about you, but I'm heading to the kitchen to see what's for breakfast, right? And the bad news, by the way, you don't smell when you're asleep. So the smell of bacon did not wake you up. Something else woke you up. That's why you need smoke detectors, right? Because you do not smell smoke when you're asleep. Dogs, on the other hand, can smell when they're sleeping and be woken up by smells. Humans cannot. Your lazy ass nose, which gives up a lot of the time, doesn't even work at all when you're sleeping. Right? If you're actually asleep, you do not smell. But I guess bacteria, you know, they can sense all the time too. I don't even think they sleep. Make sense? Right? So that's how they're sensing their environment.
Yeah, protein receptors are on the cell membrane.
In the membrane, embedded in the membrane.
Not the wall.
Yeah, embedded in the membrane. Yep. So the protein receptors are in the membrane, not the cell wall. The cell wall, remember, is just a rigid wall, right? Helps provide rigidity, keeps them from doing what? Bursting under osmotic pressure, and it helps define their shape. But stuff easily travels through them, right? There's big holes in that cell wall, right? Stuff easily travels in and out of that cell wall. What it interacts with is the membrane. Good question, Daniel. Thank you. So one other one I want to bring to your guys' attention here is this animation. Where is it? I think it's... I don't know. I thought there was one on prokaryotic movement. But this one's really cool. You can actually see the... I'll play it for a second. There's the spirochetes. Isn't they cool? That's actually under the microscope. Them twirling through. Pretty cool stuff. So the causative agent of Lyme disease, right? Borrelia burgdorferi is a spirochete. So is what causes syphilis. This is something else. Oh no, I don't know what that is, but that's pretty cool. So they literally do that in our tissue, right? So they can swim through, they can corkscrew through your tissue. The causative agent of Lyme disease does that. All right, so House is scaring me. I got to go away from him. Okay. So as you saw in that animation too, as I said, even if they're covered in flagella, they're going to orient them in such a way that it's going to allow them to propel forward in their runs. But then they kind of tumble all over the place when they tumble. They don't turn very well, right? But they can run pretty straight. And, you know, it's clockwise or counterclockwise for runs and tumbles. I never remember which is which, but I certainly don't expect you guys to know it to that level of detail. Just that they rotate. So that brings us to eukaryotics. So eukaryotic composition, structure, and function of internal organelles such as the endoplasmic reticulum, and this should be reviewed from biology class way back when, right? Things like the Golgi apparatus and lysosomes and the nucleus. Although nucleolus is probably news to you guys, right? What was that region within the nucleus? What's being made in the nucleolus? Ribosomal.
Oh, ribosomal. Through the one that transmits?
Ribosomal what?
RNA?
RNA. Everyone together. Ribosomal
RNA.
RNA. And then, of course, ribosomes are made out of ribosomal RNA and protein. And what do ribosomes make? Proteins. And then we have our mitochondria and chloroplasts. And there's a theory that goes along with these last two organelles, referred to as the Endosymbiotic Theory. And this is the theory that these two powerhouses, one that focuses on oxidative phosphorylation, right, using electrons from molecules to generate energy, as opposed to photosynthesis, which uses what as energy? Sunlight, right? But same principle, right? They're going to generate and store energy, usually in the form of ATP, right? That very high energy bond between the phosphates and that modified nucleotide we know as ATP. But when you look at these guys, you'll see that they have many membranes inside. They have an outer, in the case of this mitochondria, an outer and an inner membrane. They have their own DNA. They have their own ribosomes. And when scientists looked at the DNA and the ribosomes, they found that it was prokaryotic in nature. Not eukaryotic, but prokaryotic. Same holds true for chloroplasts. So the belief was that these were free, independent living cells all on their own. They believe the chloroplasts were descendants of what we see today as cyanobacteria, photosynthetic bacteria. They have the same chlorophyll, they do the same type of photosynthesis. So the belief was that a pre-eukaryotic cell engulfed one of these prokaryotic cells. Right? So the chloroplast was once a free living cell all by its own. And then it was engulfed by the eukaryote. And the eukaryote and the prokaryote began this symbiotic relationship. So much so that the chloroplast cannot live on its own anymore. And that cell cannot live, obviously, without its chloroplast because it's generating energy for it. So some of the abilities that the chloroplasts have are no longer encoded in its DNA that it has inside of it. It's actually encoded in the eukaryotic cell's DNA. So they can't survive without each other. Right? They began this relationship, now they're inseparable. It's kind of like what happens when you get married, right? You kind of forget how to do laundry, and the other person does it, right? So if you decide to leave that relationship, you might have to learn how to do laundry again. That's possible for us, but that relationship has been going on for so long, right? They can't exist without each other. But there's definitely direct evidence that mitochondria and chloroplasts were once free living cells all on their own. So mitochondria are powerhouses, right? I thought I had descriptions under these. And of course, chloroplasts are for photosynthesis. Oh, that's what I want to say. So your mitochondria, you get from your mom. They're in the egg. Sperm are basically literally just mobile DNA. Like all your organelles you get from your mom. But the mitochondria are very important in that, like I said, they have their own DNA and certain abilities all on their own. So you can unfortunately inherit genetic diseases from your mom related to your mitochondria. So I had a friend in college, he was losing his eyesight because he got defective mitochondria from his mom. The weird thing is that for some reason for men they're affected. Now the sperm has a few mitochondria because obviously it needs it for the energy to move its flagella to get where it needs to go. But usually they don't make it into the egg. And again, there are not enough of them to make a difference. So all the mitochondria you get are from your mom. And so if there's a defect, it's directly from your mom. So in his case, it was directly from his mom. And his sister, although she wasn't going blind, because of that information, decided not to have children. Because, again, all her mitochondria are defective. She passed them on to her children. There's a chance that they would develop this disease. So she had cats instead. Right? She didn't want to have a son, and for some reason, for males, the disease tends to show itself and progress more than for females. So although she wasn't going blind herself, she could give birth to children that could potentially go blind later in life. I don't remember the exact name of the disease anymore, but I could tell you if you want. I can look it up. You could look it up even. Blindness associated with mitochondrial defect. So, make sense? All right, so moving on. So, this ability that's eukaryotic only of being able to engulf things, to take the membrane and surround it, is referred to as endocytotic pathways. And there are some that are specialized. Endocytosis in general is just taking in really large stuff using the membrane to encase it within a vesicle. Phagocytosis is a little more specific. We're taking in very large components, even whole bacteria and viruses. And we have specialized white blood cells in our body that we call phagocytes, because that's their main job, is phagocytosis, to engulf bacteria, viruses, things that get into our body that could harm us, and to destroy them. Autophagy, on the other hand, auto means what? Self. So again, those mitochondria and chloroplasts and things like that that need to be recycled or digested within, we want to do that safely. We want to do it within a vesicle. So all these processes start by creating a vesicle, whether it is taking stuff from outside or putting stuff within our cells into a vesicle. Then the next step is going to be digestion. And again, we want it to be controlled digestion. So we're going to do this within a vesicle, within a membrane sac, within the cell. We have special vacuoles are called lysosomes. They specifically contain digestive enzymes. Digestive enzymes we don't let run willy-nilly in our cell because they would just eat it up. Okay? Just like the enzymes in your stomach, we have a very thick layer of mucus that protects our stomach from eating itself. Right? If we didn't have that, that's when you end up with ulcers, because your stomach starts eating itself. You produce hydrochloric acid in your stomach and proteolytic enzymes, pepsinogen, to digest proteins. You would digest your own cells' proteins if it wasn't for that very thick layer of mucus that you make to protect yourself. So for inside of our cells, we've got to protect ourselves as well, and we do that with membranes. Right? We create these vesicles, these membrane sacs within the cell, and that's where we do the digestion, in that controlled, sequestered environment within the cell. So what organelles are responsible for these pathways? Well, you definitely need lysosomes. And how are lysosomes made? Right? So that's covered in this animation. Right here, formation of lysosomes. But I can't get my audio to work, but that's okay. So inside of those digestive enzymes, hydrolytic, meaning they use water to lyse, but it all begins here. Anybody know what this organelle is? The rough endoplasmic reticulum. It's labeled. They don't have the rough part. What's the rough? What are these? Ribosomes. Ribosomes make what? Proteins. Most of their enzymes are proteins. So they were made here, right? And the vesicles traveled here. What was this? This is a Golgi apparatus. My nickname for this guy is the packing plant, right? He packages stuff up and sends it on its way. In this case, it sent it to another vesicle to digest it. So here they actually showed, let me rewind. Let me freeze it. So stuff coming in from outside, and this looks like bacteria to me, right? Little rod-shaped. So that we form this vesicle. Here we've got the lysosome, and they're going to fuse to each other. So again, we're doing this digestion safely within that vesicle. Just play. There we go. It's being digested. And then they'll even, spit out the trash. You'll see this fuse with that membrane and spit it out. Here, it wouldn't attack, the lysosome wouldn't attack the mitochondria. So that's autophagy, right? We destroyed something from within. All of those are endocytosis, right? You're doing it within, endo within those vesicles. Oop, wrong button. So ER stands for endoplasmic reticulum. It can be smooth, which means it doesn't have ribosomes, and in that case it's mostly lipid synthesis, fats and stuff. Your rough ER is making proteins because of the ribosome component. Who would I call the packing plant? The Golgi apparatus, right? So things that come off the endoplasmic reticulum and either the rough or the smooth, a lot of times they're going to be packaged and processed even further by the Golgi apparatus. So what actually comes off of the Golgi apparatus is referred to as a lysosome, right? So maybe in the rough ER they made one type of enzyme, right, and brought it into a vesicle and sent it to the Golgi apparatus. And then another vesicle contains a different enzyme. And the combination of those two vesicles is what happens in the Golgi apparatus. So that what finally leaves the Golgi apparatus is what we call a lysosome. It doesn't just have one type of enzyme, it has multiple enzymes packaged in that vesicle. So a lysosome is a membrane-bound acidic site of intracellular molecular digestion. We're literally tearing up proteins and lipids and such as that. Mostly protein digestion, though. So when we say phagocytosis, we're talking about a specific type of endocytosis. We're talking about the type where we're taking in large things like bacteria. And so that vesicle that engulfs the bacteria is commonly referred to as a phagosome. But again, it's going to fuse with the lysosome and be destroyed. A lot of times when we just take stuff in by endocytosis and put it into a vesicle, we may need to digest it using a lysosome. And then we can even have lysosomes fuse with mitochondria, large components within the cells, to be able to digest them safely within the cell. And since that's within the cell, something from within the cell, it's referred to as autophagy. But it's still endocytosis, it's still digestion within the cell. So this chart from your book does a good comparison of prokaryotes and eukaryotes, and even does the division between the two major types of prokaryotes, the two domains of bacteria and archaea. But it goes into a little bit more detail than I did in my lecture, so you'll notice I've scribbled some lines through those added details that you don't have to worry about. So again, very similar to that first Connect assignment that we were reviewing earlier, but then added in some additional details. Like ribosome size, remember 70S is prokaryotic, where 80S is eukaryotic. That peptidoglycan is a defining characteristic of just bacteria. They're the only ones that have that composition to their cell wall. Endoplasmic reticulum, Golgi apparatus, lysosomes, those are all organelles, right? So they're only found in eukaryotes. Cytoskeleton does exist in prokaryotes and eukaryotes, but it's very simplistic, right? Where the cytoskeleton for eukaryotes was much more complex. Can anyone name some of the components of that? Microtubules, yep. The microfilaments. One more. Intermediate filaments. So remember, it goes microfilaments are the smallest, and then your intermediate filaments, and your microtubules are the largest. And your microtubules are very important for eukaryotes because they also make up an external structure that helps in locomotion, and that's what? The cilia and the flagella. What's the difference between flagella and cilia? So only eukaryotes have cilia. But what's the difference between a eukaryotic flagella and a eukaryotic cilia? They both wave. For eukaryotes, flagella wave, cilia wave. Short. Cilia are short. Flagella are longer. And a cell that has cilia, usually, I mean, you know, you could survive with 1 or 2 flagella, right? With cilia, because they're so much shorter, the cells tend to be covered in cilia. So think of, in that case, remember paramecium, right? Completely covered. We have ciliated cells, right, in our respiratory tract. They move, not the cells. The cells are stuck, but what do the cilia move? The mucus you produce. Yep. They beat and move the mucus that you produce to help coat and protect the lining of your respiratory tract. But again, it takes a lot of them all beating together to move that stuff. So cilia are unique to eukaryotes, right? Prokaryotes, bacteria do not have cilia. They have flagella, and theirs even move differently, and structurally they're different than bacteria. Bacteria and archaea are different. Even though they both have flagella, they're structurally different and they move differently. So, huh? No. So his question is fimbriae, which are bacteria, protein appendages, are they similar to cilia? No. They can be used for locomotion, right? But they're mainly for attachment. Whereas cilia, 100% for locomotion. Whether it be the organism itself or in the case of us with our mucus, to move the mucus. So another one, they go into a little bit more detail. Well, here's the flagella one, right? Remember, these are teeny tiny. For bacteria, prokaryotes, we'd have to do a special stain to see them. They're submicroscopic. They're so tiny. And it's just a single protein that makes up those flagella. Wherefore, as we said, with eukaryotes, the flagella is much more complex. And usually, under certain circumstances, you can actually see it under the microscope. And it's made of microtubules. And it's in a particular pattern, you'll see. Bunches of 9 around the perimeter and 2 in the center. So it's said to be that 9-2 pattern. Which I had a picture of that, right? I don't remember if I talked about it. There it is. There's the picture. So you see the microtubules, the circles, and then there's 9 pairs around the edge and then 2 in the center. Where for prokaryotes, it's just this one protein fiber. It's all one protein stacked up. That's it. Flagellin protein. That's it. We're much more complex when you get to eukaryotes. And then, of course, the eukaryotic structure that's similar to flagella that prokaryotes don't have are cilia. Right? Cilia. Completely covered in cilia. They're just shorter. They move the same, they're just shorter. They wave back and forth. So do the flagella for eukaryotes. Now, another detail that they go into here is the differences in the bonding of the plasma membrane. So notice that it's an ester linkage for bacteria and archaea, but it's a diether linkage. I don't expect you guys to remember that chemical level of understanding. The important thing here is, remember how we talked about differences between bacteria and archaea? one of the major differences that allows these archaea to survive in these extreme environments are those structural differences in their membranes. So in addition to this different linkage, what else did we say that archaea can do with their membranes? They can form those monolayers. They have the branching to the fatty acids. One more. They can cycle, right? So they can actually change the width of that membrane, right? They can cycle up, form those cyclic bonds, those pencyclics, 5 carbons circled together, and shorten the fatty acids, so actually shorten, make their membrane thinner. Interesting little guys they are. And then the last one down here, they have gas vesicle. This is actually an example of, we just talked about just in general inclusion bodies, right? Where they'll package stuff inside. A gas vesicle is an example of an inclusion body, which remember are just prokaryotes, right? Have inclusion bodies. And that's where they're surrounding the stuff. Remember to take it out of the equation of osmotic pressure, right? And in the case of gas, that'll help them with buoyancy too, if they're in an aquatic environment. So I bet you're like, woohoo, early day, right? It is going to be early, but I have one more thing I want to talk to you guys about. Okay. So, and I'm still building it, so I didn't turn it on. So we have our test 1 coming up on Tuesday, covering this module that we just finished. There's the lockdown browser if you want to download it so that once the test is available, you'll be able to view it from home. Close the door for me because I'm not done and I don't want to listen to them and stop packing up. Because then I can't hear. We'll have another unit, right? Another module. If you look at the syllabus, if you actually look at my syllabus, actually I can pull up my syllabus addendum so you know what's coming and what to expect. So here we go. So module 1, right, which we just finished, getting ready to have our test. Module 2, right, which I'll start posting stuff for you guys, so as soon as you get done your test, you can start looking ahead. We're going to cover viruses and genetics and growth and control. And notice some of these chapters are in parentheses. That's because we don't cover the whole chapter. In the case of growth, one of the major things I talk about, which is nutritional types, for some reason they moved it to chapter 11 on me. So that's why that's in parentheses like that, matches up with that. And then the next module 3, it looks like, woo, easy, right? No, these are 2 monster chapters on immunology, like monster chapters, right? And then we have module 4. So typically in the past, we do research projects and stuff, which, you know, some people like, some people don't like, right? But since a lot of you guys are going into the health field, what I've done is I've changed it up and I do case studies for this module. So instead of giving you guys a research project, we do case studies. So you still have to do some reading, right, and researching and answering questions. So I started building that module. I'm almost done. So if you want to get started working on it, you have all semester long to work on it, right? But you have to complete it by the end. And what I do is, and I'll turn it on, but I'm still building and adding instructions. This first case study on Listeria, these are the questions that I want you to be able to answer from reading. This is the case study. And this is even a link to a book that does a really good job at covering that disease. All right, so you really don't have to go anywhere other than on Canvas. Then there'll be a link below this where you're going to answer questions based on these questions that you should have read and tried to answer on your own. So it's going to be like an in-Canvas quiz, where I think it's 20 points for this class. Don't quote me on it, though. I have to look at my grading scale. And then once you get done, it will tell you the answers. These case studies, there's 5 other case studies. So here's the case study itself from the case study book. Here's information from a book and another book. And then here is a PowerPoint of me going through all the answers. Most of the answers, I should say. I have recordings. I used to do these in class. I have recordings of me giving these lectures as well that I'll post for you guys. What you want to do is gather all your material. You may even want to print it out, right? Go through, read up on all this material, organize it, and then at the end of this unit, I'll have a test that you can do at home. You don't need the lockdown browser or anything. It's right there in Canvas. It's open notes, right? You do it at your own pace, but you can only open the assignment once, right? And that's going to count as a test grade, as your project grade, as your Module 4 grade. It's 100 points. Make sense?
But I have one question. So can we print out?
You can print everything from this module that I post.
You said there's 5 of them?
Yeah, I think there's one more I haven't posted yet. There's 6 total. Listeria, I don't give you the answers. I don't give you the answers until you take the quiz on it. There's fungi, so that's 1, 2, 3, 4. I didn't put a title for meningitis yet. I got to add it in there. And then there's Listeria, shigellosis. I got to add one more. There's 6 total. No, the test is on all 6. You got to read up. That's why I said work on it throughout the semester, you know. And in fact, I'll refer to these sometimes. So like we've got viruses coming up. What is hepatitis caused by? Yeah, hepatitis means inflammation of the liver, which can be caused by several viruses and one viceroid. So I would suggest if you're going to pick, I'd read hepatitis one first because I will actually talk about it when I talk about viruses. And so you'll have some familiarity, right, with that particular virus that'll help in your understanding.